Okay, here they have given microsporogenesis. The microspore, as they are formed, are arranged in a cluster of 4 cells. I told they forms like this. They forms like this structure, which is known as the cluster of 4 cells. 1, 2, 3, 4. Okay, so 4 is there. So microspore, they have given the microspore as they formed arranged in the 4 cluster of cells. Okay, the microspore tetrad, this is also known as the microspore tetrad. It will be the microspore tetrad. Microspore tetrad, it will be always haploid because it is derived from the PMC, pollen mother cells. Okay, that will be the diploid. So microspore tetrad will be the haploid. Okay, N. Now, as the anther mature and dehydrate, when the anther mature and dehydrate, the microspore dissociate from each other and develop into the pollen grains. Now I told it will undergo this microspore tetrad, single microspore undergo the mitosis. It will undergo the mitosis. Okay. After mitosis, they will form single microspore. They form the two celled structure. That will be single, but they have the two celled structure. One is called, which is the large, it is known as the vegetative cell, and another is the small, which is known as the generative cells. Okay, vegetative cells and generative cells. Okay, so now it have given, read it. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released with the dehiscence of anther. Okay, they are released with the dehiscence of anther. Now pollen grain. The pollen grains represents the male gametophyte. Very, very important. This is the MCQs, PYQS. The pollen grain represents the male gametophyte. Can you tell which structure represents the female gametophyte? Anyone, can you tell which structure represents the female gametophyte? Anyone, do you know? Which structure represents the female gametophyte? That is embryo sac. Embryo sac, which represents the female gametophyte. Pollen grain represents the male gametophyte. Is it clear? Is it okay? Shreya?
Yes, sir.
Very good. The pollen grains represents the male gametophyte, while embryo sac or embryo sac, which represents the female gametophyte. Okay, now if you touch the opened anthers of hibiscus or any other flowers, you would find deposition of the yellowish powdery pollen grains on your fingers. Okay, that if you touch this anther, if you have seen hibiscus roja sinensis, that you can say China rose. If you touch the China rose of anther, then you will get the powdery some substances. Okay, and sprinkle these grains on a drop of water taken on a glass slide and observe under the microscope. They are saying you can take this pollen grains and the structure of the pollen grain you can observe under the microscope. So now you can see the microscope, you will get the different types of the structure they have given. Like here one here another pollen grain, another pollen grain, this pollen grain, different. That scanning electron microscope means this structure generally observed by the SEM means scanning electron microscopy. Scanning electron micrographs are same TEM. Transmission electron, that is not up to your level. That this microscopy, this is very high level. But what I'm saying, just look, scanning electron, that you can say in short SEM. So with the same scanning electron microscopy, they observed this structure of the pollen grain. Okay. Now, a microscope, you will really be amazed at the variety of architecture, size, shape, color, designs seen on the pollen grains from different species. If we take from the different anther of the different flower species, you will get the different type, various type of the shape, size, color. Everything is going to vary according to their species. Okay. Now, pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. The diameter of pollen grain will be the diameter of pollen grain will be the 50 to 25, means pollen grain, it will be the 25 to 50 micrometer in diameter. Clear? So the diameter of the pollen grain, it will be 25 to 50 micrometer. Again, it has a prominent two-layered wall. Very, very important. Now one question, that is PYQS, they have the prominent two wall. It has that prominent two-layer wall, the hard outer layer called exine. The hard outer layer, which is known as the exine, and it is made up of the sporopollenin. Very, very important. This is the PYQS. Outer layer, suppose that I'm drawing the structure of the pollen grain. The outer layer of the pollen grain, that look it here. The outer layer of the pollen grain, which is known as the exine. And this exine is the hard structure. And these exine generally made up of the sporopollenin. So these exine, which is made up of the sporopollenin substance. Okay, sporopollenin substance, and this sporopollenin is the hard structure, means no one enzyme can digest this structure. Are you getting what I'm saying? Sporopollenin, which is found on the outer layer, means it is the hard protective structure of the pollen grains, and no one enzyme now discovered which have ability to degrade this structure. That means exine. So exine structure cannot be digested by any enzyme. Is it clear? Is it okay? Is it clear to all of you?
Yes, sir.
Shreya?
Yes, sir.
Very good.
Yes, sir.
So outer layer of the pollen grain, it is made up of the exine, and this exine, which is made up of the sporopollenin, and no one substance can digest this structure of the sporopollenin. Now look at here, which is one of the most resistant organic material known. It can withstand high temperature and strong acids and alkali. Means the pollen grain, they can sustain for the long time. We have already studied in evolution. You will also study that panspermia theory. For the origin of life, they are saying one theory which is known as the panspermia theory. In the panspermia theory, they are describing some sperm, some pansperm like some sperm like structure. They came from the another planet, and they came from the another planets and on my planets or our planets, and they form a new life. Okay, that is panspermia theory. That means you can say that will be also spore like structure, because they have ability to resist high temperature, acidity, alkali. So pansperm, that is the spore like structure which have ability to generate. Are you getting what I'm saying? So this pollen grain, so pollen grain outer layer, which is made up of the exine, and this exine is made up of the sporopollenin. And sporopollenin, they have ability to, it is the resistant organic material. This is the resistant organic material. It can withstand high temperature and strong acids and alkali. Okay, now that sentence which I told to you, here they have given no enzyme that degrades the sporopollenin is so far known. Till now unknown. Okay. Pollen grains exine has prominent aperture that called the germpore. Okay, that called the germpore. Suppose that I'm drawing the structure. This is the pollen grain. Everywhere what you will get, look at here. Everywhere they have there is the structure which is formed. But some places this structure is not present. Okay, some places this structure is not present. Now here they, one second. Yes. Okay, that means you can say they have how many pore? They have the 3 pore. Look at here, pore number 1, Irandu and Moon. They have the how many pore? 3 pore. So this pore is also known as the germ pore. This pore, wait, I will change the color because color is not contrast. Okay, this structure is known as the germ pore. This structure is known as the germ pore, or simply it is also known as the colpet. This is also known as the colpet. Okay, in dicot, it will be the tricolpet. In dicot, tricolpet. If the dicot pollen grain is there, so they have the 3 germophore. Just try to understand what I'm saying. If the dicot pollen grain is there, so these have how many germophore? 3 germophore. That's why it is called the tricolpate. First type of the pollen grain which have the 3 germophore. This is called the tricolpate. Okay. In case of the monocot, they have the single germophore. Monocots, it always bears only the single germophore. Suppose that I'm drawing this structure like this. This is the exine. They have the only single germophore, and this single germophore also known as the monocolpate. Okay, they have the only one structure, one pore, which is known as the monocolpate. Okay, so monocolpate and tricolpate. Tricolpate found into the dicot, and monocot have the monocolpate. Monocot, monocolpate. In this way, you can learn. Colpate also known as the germophore. Now, okay. Look at here. Pollen grains exine has prominent aperture that is called germophore, where the sporopollenin is absent. There is no deposition of the exine, no deposition of the sporopollenin. Pollen grains are well preserved as fossils because of the presence of sporopollenin. I told the pollen grains you can also preserve for the fossil purposes. Our fossils we have to also found different. The exine exhibits a fascinating array of patterns of designs. Why do you think the exine should be hard? That will be why they have nature provided for the sustainability, for the durability. The exine it made up of the sporopollenin. Okay, what is the function of germophore? Germophore, by which there is the formation of pollen tube takes place. Germophore is the site from where the pollen tubes takes formation of the pollen tubes takes place. Now look at the inner wall of the pollen grains. It is made up of the intine, and it is a thin continuous layer made up of the cellulose and pectin. Inner wall, which is made up of the cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grains mature, it contains 2 cell vegetative cells and generative cells. I told already that microspore, when the microspore, when the microspore form, this microspore undergo the mitotic division. That will be microspore already haploid. I told when they form the microspore, PMC, pollen mother cell, and microspore mother cell, that would be diploid. After when they are undergoing the meiotic division, they will form microspore, and each they form the 4 different microspore, 4 different microspore, and each microspore will be the haploid. Each microspore, they have the 4. In one of the 4, clear, out of the 4, one of them they may be enter into the mitosis, or all of them is 4 direct can enter into the mitotic, or one of them they can enter into the mitosis. That is not particular criteria. Okay, so 4 microspore is forms, each are haploid. In out of 4, one of them they undergo the mitotic division, or 4 microspore they undergo the mitotic division. Okay, when enters into the mitotic division, I told, suppose that this is the microspore. This is the microspore. When enter into the mitotic division, there is the nucleus is going to divide. They form the 2 different cell. They form the 2 different cells. One here and here. Okay, they have the 2 different cell, but cytoplasm will be same. Now you can say the cytoplasm will be cytoplasm. It's not going to differentiate. They have the same cytoplasm, but they have the 2 different types of the cells. Okay, the cells which will be big in size, the cells which will be big. Now you can see they have given the cells, the size will be big. This is called the vegetative cells. Now vegetative cells is large in size, vegetative cell, and those cells which is small and found at the base, this is called the generative cells. This is known as the generative cells. And which is too much important, generative cells is very important, because generative cells undergo again mitotic, and they form the 2 male gametes. They form the 2 male gametes. So in this way, which cell is very much important? Generative cells, because generative cells undergo the vegetative cells and generative cells. Now you can say here vegetative cells and generative cells. The vegetative cells is bigger as has abundant food reserve and large irregularly shaped nucleus. The generative cells is small and floats in the cytoplasm of the vegetative cells. Means I told the cytoplasm is not divided. The cytoplasm will be the same, but they have the two different types of the cells. Okay. Now, uh, generative cell, it is the spindle shapes with the dense cytoplasm and a nucleus. Now, the shape of generative cells, it will be looking like as, that is the spindle shape. The structure of generative cells, it is looking like as a spindle shaped structure. A spindle shaped structure. Okay. So spindle. In over 60% of the angiosperm, pollen grains are shared at the two cell distance. Very, very important. This is the PYQS, previous year question. What is saying, just read this sentence. It is saying, in most of the angiosperm, how many percent? 16. It over 60% of the angiosperm, pollen grains are shared at the two cell stage. Pollen grains are shed at the two-celled stage. Where shed? Shed on the stigma, shed on the plant, shed on the flower. Okay. So shed at the two-celled stage. In the remaining species, the generative cells divides mitotically to give rise the two male gametes before pollen grains are shed. That is three-celled stage. Okay. Remaining means 40%. In the 40%, what is saying? In the remaining, that means 40% species, generative cells divide mitotically. I told this generative cells also divide mitotically, they form. Mitotically to give rise the two male gametes before pollen grains are shed. That is three-celled stage. So 60%, 60% two-celled stage it is going to shed, and 40% it will be three-celled stage shed. Okay. When it will arrive into the three-celled stage means when generative cells again enters into the mitotic division. Is it clear now? Concept is clear. Is it? And bronchial affliction in afflictions in some people often leading to the chronic respiratory disorder like asthma, bronchitis. Okay. Some persons we have to see they have allergy from the flower. If you have seen such type of the flower, or some person also they have the allergy from the pollen grain. If they inhale this pollen grain, they can cause such type of the disease, maybe bronchial afflictions, or they can also cause the chronic respiratory disorder such as asthma and bronchitis. Bronchitis means swollen into the bronchi. Inflammation, tis. If anywhere came tis term, if it will come, that means inflammation of the bronchi. Inflammation of the bronchi, that is called the bronchitis. Bronchitis, arthritis. Okay. Arthritis means inflammation in joints. That means if anywhere tis come, that means you can say inflammation of the particular part. Okay. Now, so this is the chronic respiratory disease also caused by this pollen grains if those person which have allergy and inhale this pollen grain. Okay. It may be mentioned that Parthenium. Parthenium, this is also called the Congress grass. Parthenium, it is also called the Congress grass. Why we are saying Congress grass? Because it came when we bought from the wheat, when we bought wheat from USA. So that time it came as a unwanted weed. Are you getting what I'm saying? I'm saying that when we bought wheat from USA, so that time along with wheat, some weed means that is waste substance. Along with wheat grain, some weed of this Parthenium grass came on our India. Okay, came on in our India, and because this is they have too much ability to occupy maximum space, so the growing capacity is high. Okay, so Parthenium or Congress grass, or it is also known as another name of this grass, which is known as the carrot grass. So three name: Parthenium or Congress grass or carrot grass. That came, they have given, that came into India as a contaminant with imported wheat. I told when we bought from USA, when we bought wheat grain from USA, along with wheat grain, some unwanted this weed, Parthenium or Congress grass or carrot grass, it came in our India. Okay, and now they have occupied maximum space in our India because they have the too much ability to occupy the spaces. They have the this is the weed. This is the weed. Unwanted. It is the growing. Okay, now, and it has become ubiquitous in occurrence and cause pollen allergy. Okay, this carrot grass or parthenium, this also cause the whatever they have given. Pollen grains are rich in nutrients. Okay, they are saying pollen grains also rich in nutrients. It has become a fashion in recent year for use of pollen tablets at food supplement. Recent means now we are using this pollen grains as a fashion. Fashion means as a food supplements. Those persons who is doing the gym or we who is very sincere for your health, they are using such things. Okay, so now fashion now is become a fashion. Recent years we are using the tablets or food supplements which is made up of the pollen grains. Okay, now in western countries means USA and other things. Okay, USA, England and other. So western countries allows number of the pollen products in the form of tablets and syrup are available in the markets, but in India it is not possible. They are selling, but it is less in quantity. In western countries they are selling in huge quantities. Pollen consumption has been claimed on increase the performance of athletes and race horses. Okay, sometimes this pollen grains can also enhance the your energy level. Okay, that you have to seen one story, that is dope test. Anyone, can you tell what is the dope test? Dope test, anybody, do you know about dope test? Ritarani, Nandana, anyone, do you know about dope test? Anyone, can you tell, do you know about dope test? I think you don't know. Okay, leave it. Dope test means, suppose that if the person is going to play the game, suppose that India and Sri Lanka, both cricketers, they are going to play the cricket. Okay, so before playing the cricket, the doctors, they will assess, they will take their sample, sputum, urine, and sweat, and other things. They will take and check the samples whether they have taken any drugs or any energetic drugs which can enhance the body activity. Sometimes what they did, some person they took the drugs by which they can enhance their energy, enthusiastic. That means you can say they increase their energy level, and for the 3 to 4 hours they can play too much. They can play huge, means they can play for the long time. That time they will never feel the tired. So that is called the dope test. If the before 1 or 2 hours, if they are going to play, the doctors will check whether the persons took the drugs or not. That is called the dope test. Okay, whether its persons have the drugs or it had the drugs or not, that is performed through the dope test. Now dope test, it is clear. Is it clear to all of you? Shreya?
Yes, sir.
Okay,
Yes, sir.
okay, clear. Now, so this pollen grains, they can also, that has been claimed to the increase the performance, means that you can say this act like the drugs. It can increase the performance, means it can act like the drugs. So performance of athletes and race horse. So sometimes if the person it is going to play in the race horse or athletes, they are going to do your work. So that time they can use this pollen grains, pollen consumption. Okay, this is also act like as a drugs. Is it clear? Is it okay? Is it fine?
Yes, sir.
Very good.
Yes, sir.
Okay, now,
Yes, sir.
okay. Again, look at here. When the pollen grains had to land on the stigma. This is called the pollination. When the pollen grains land on stigma, this phenomenon is known as the pollination. So simply you can say, when they are shed, pollen grains have to land on the stigma. Before they lose viability, they have to bring about fertilization. Okay, they lose viability and they bring to bring about the fertilization. Means they are ready for the fertilization phenomenon. How long do you think the pollen grains retain viability? They can retain for the that will be viability is very big, because I told they have that pollen grains, they are made up of this sporopollenin enzyme. So the pollen grain viability is very much. If they arrived onto the stigma, that will be go for the fertilization. Otherwise, it will be remain for the long time, because that outer layer exine is made up of the sporopollenin, and no one enzyme can digest this sporopollenin. So due to this, the viability of the pollen grain will be very high. Okay, now the period for which pollen grains remain viable is highly variable and some extent dependent on the prevailing temperature and humidity. I told they have given the period of which pollen grains remain viable is highly variable. To some extent depends on the prevailing temperature and humidity. It depends on the temperature and humidity where they are leaving this pollen grain. Okay, look at some example they have given like that. Just look at. In some cereals such as rice and wheat, the pollen grains lose viability within 30 minutes of their release. Okay. It is saying in case of the rice and wheat, if the pollen grains shed from the flower, it will lose their viability within 30 minutes. Okay, this is the very important. This is the PYQS, previous year questions. The viability of the wheat and rice. Okay, the viability of wheat and rice grains. It will be how many time? That will be half an hour, 30 minutes. Okay, it will be the half an hour. So the time of viability of this. Okay, the time of viability of this pollen grains means the pollen grains of wheat and rice. It will be the 30 minutes of their release. And in some members like Rosaceae, Leguminaceae, and Solanaceae, they maintain for the month. Okay. So in case of the Leguminaceae, that is the pulses. Rosaceae, rose, and Solanaceae, that means Solanum nigrum tuberosum. Okay. Solanaceae, that this family also have given Leguminaceae. No, Leguminaceae is not there. Papilionaceae, Solanaceae, and one more also. Papilionaceae, Solanaceae, and Liliaceae. So Papilionaceae, Solanaceae, Liliaceae, these three family we have studied into the +1 standard. Okay, you may have storing semen spawns for many animals, including human, for the artificial insemination. For the artificial insemination, we have to see. You have already seen in your rural areas, the doctors they took the big nitrogen cylinder. In the nitrogen cylinder, they put the sperm, and these sperms used for the artificial insemination. Okay, this is sperm basically used for the artificial insemination. So these sperms they have the too much viability. Why? Because they put into the nitrogen cylinder, and nitrogen gas they act as a cryopreservative. They can preserve for the long time. Okay, so now what is saying, just read this. They have given, you may have heard storing semen sperm for the many animal, including for artificial insemination. It is possible to store pollen grain of a large number of this species for a year into the liquid nitrogen, and the temperature of liquid nitrogen is the -196 degrees centigrade. Such stored pollen can be used pollen banks similar to seed banks and programs. Okay, so means -196, that is the temperature of liquid nitrogen. Okay, so this is the overall story about the microsporogenesis and pollen grains. Remaining portion we will talk into the next class. So thank you.
Thank you.
Thank you, sir.
Thank you, sir.